Tonight, on After Hours with Rictor, my co-host Tammy is the only one that can summarize what's about to go down. Tammy?
It's Judgment Day for Melba.
Welcome to After Hours. My name is Rictor. I am your host.
Rictor!
What?
Leave him alone!
It's Judgment Day for Sasquatch.
Thanks for dropping me like a stud.
Now we got Rictor going. We're gonna have to hear about it all night.
That's a bunch of screaming memes out there, and that's the scoop that has been reported so far.
By your opinion that you are no-kill, you are dooming the species to be extinct.
Well, when you don't believe in Bigfoot, everything you see that might be one is something else.
We thought that we had the holy grail of DNA.
When are we going to make a video, Rictor? And I mean not an X-rated one.
Dr. Todd, you've also been called the scoff dick.
Yeah, well.
You can't talk about
I can't!
Hello? Is this thing on? Am I muted? Can you hear me?
Hey, Rictor, I've got a question for you. How does it feel to lose Bigfoot Bounty?
My question is, why do you think Bigfoot is real?
Justin, we need a body to give Dr. Disotell.
Oh, I'm on it, Rictor.
Dude, you're missing.
Hold on. I almost got it. I almost got it.
Justin, she's getting away.
Shut up, you're gonna make me miss my shot. Shut up.
Oh man, we almost made MK Davis happy. Tonight on After Hours with Rictor, we have a first, two doctors. The first one, Dr. Haskell Hart. Now, if you've been following the Bigfoot community, you know, he kind of shook things up for the Ketchum camp about a month and a half ago. Well, we're really glad to have him on to explain his DNA findings in regards to the elusive mythical creature known as Bigfoot. Dr. Hart received his bachelor's in science in chemical engineering with the highest honors and distinction in curriculum from the University of Illinois. He holds his MA and PhD degrees in physical chemistry from Harvard University, and his major professional experience has been physical, inorganic, and analytical chemistry. Now, a lifelong amateur naturalist, Dr. Hart has followed the Bigfoot Sasquatch news since the Patterson-Gimlin film. Following the publication of the Ketchum DNA studies, Dr. Hart immediately began to examine those results and conclusions. His internet paper, quote, "Distinguishing Related Species from Incomplete NU DNA," unquote, is the result of 8 months of education, study, and original research. Now, Dr. Hart entered the Bigfoot scene early in 2013 when Dr. Ketchum announced his support of her findings that Bigfoot DNA shows these beings' mitochondrial DNA to be from human females and the nuclear DNA to be from an unknown species. And of course, we all know that she believes this unknown species to be, quote, "not of this world," unquote, and that she has called them angels. Angels. Now, however, at some point after that, something changed in terms of Dr. Hart's support of the Ketchum study. Now, returning to After Hours is my buddy, my DNA teacher, Dr. Todd Disotell from Spike TV's $10 million Bigfoot Bounty. And he has a few things to say in regards to this whole Ketchum study. So, welcome, gentlemen. Dr. Todd, tell us about you. You are a professor of what at the New York University?
I'm a professor of biological anthropology in New York University Center for the Study of Human Origins, and I specialize in what I refer to as molecular primatology, the study of DNA of primates, including humans, being that we're just a very peculiar primate, some of us more peculiar than others. PhD and master's from Harvard as well. So it's a pleasure to meet you, Dr. Hart. I read your paper with great pleasure.
Well, likewise, sir. I've seen you on TV, of course, and on these podcasts, and it's a pleasure to be with you, and you can call me Haskell.
Okay,
and,
thank you.
I'll call you Todd.
That's fine.
Joining me is my beautiful, sassy Tammy Murray, my co-host from the very beginning.
Oh.
Tammy, you've got something there with you in paper form. What you got?
I have a bunch of pages that I printed off, and this is the paper by Dr. Haskell Hart. And I've been reading the good parts, and like I was telling Dr. Disotell earlier, there's some parts of it that really bother me about your paper, Haskell, and it's, I don't know if you'll be able to see it. That stuff right there.
Just a
That's just formulas.
I just put it there to confuse people.
I see stuff like that, and it makes the hair stand up on the back of my neck.
Well, that's why we have these two gentlemen here to explain it to us, bring it down to us, because I'm just an artist and webcaster, and Tammy is a Bigfoot glass pendant maker and webcaster, and the two of us do not have anything in our background in regards to science and understanding DNA. I know how to collect DNA, so does Tammy, but understanding and breaking it down is where these two fine doctors come in. So welcome to After Hours, guys.
Thank you.
Now, Todd, what first got you interested in Bigfoot?
I was asked, oh geez, it was a long time ago, well, a decade ago or so. I was actually asked by Nat Geo to the National Geographic Channel to analyze some samples. And I believe the first ones I did were some that my friend and colleague Meldrum had collected. And I did a few shows for Nat Geo as well as Monster Quest on History Channel, analyzed sort of one-off analyses of various samples of Bigfoot, Yeti, Yowie. So I've done some Australian ones. I've done some from the Himalayas. Turned out to be mountain goats. Imagine that. Hair found high up in the Himalayas from mountain goats. I've always been willing to work with people like Meldrum and other sort of credible, or at least semi-credible people to do these analyses, because it's exactly the work we do day in and day out in my laboratory. I mean, we analyze biological samples of primates to identify what species, subspecies, population, or whatever they are. And so none of the techniques in so-called cryptozoological molecular work are any different from what we do every day. And, you know, it's fun. You know, as I keep telling people, if what you're doing isn't fun, why should you be doing it? So, but I also get to educate the broader public about science, evolution, DNA, the scientific method, just skepticism. You know, the what ifs, what else? I mean, what else is more important than what is or what if? So my research group and laboratory has been involved in the description of multiple new species and subspecies of monkeys and apes over the years. We're working on some lemurs right now, trying to figure out if they're a new unknown species or heretofore unknown species or not. So same thing, same samples even. Hair, shit, saliva, blood, all the same stuff. Standard scientific research.
Okay, Dr. Hart, first of all, how did you get involved in all of this Ketchum studies, and why did you originally support her findings?
A good question. Well, as you've read, I am a lifelong naturalist, so I have kind of followed people's reported findings or sightings of Bigfoot since the Patterson-Gimlin film back in the 60s. And, of course, you know, as a physical chemist, I must say first that I'm not a biologist, I'm not an anthropologist, or a geneticist. But as a physical chemist, I do understand how DNA works. I'm not a biochemist, but I understand that much about it. And I was fascinated that these sequences were reportedly, the 3 nuclear sequences, reportedly from an unknown species. So I thought, well, I'm retired and I'm trying to keep the brain alive, so why don't I just look into this a little bit and try to learn some things about how people sequence DNA and how they match it against known sequences. And I got in, and my preliminary results were partly wrong. I did think that these sequences had a lot of homology to humans. And then I found a paper that was written about the panda genome, and 60% of human, panda, and dog genomes are the same at the 90% ID level. And so that makes things a little complicated. You can't just look at a number and say it matches 97% and therefore it must be the same. You've got to compare that to what else it matches. That's what I found fascinating here, and I learned a lot about the NCBI databases, which are not linked, and you have to search more than one to get your answer sometimes.
So was this your first red flag?
Well, I'll tell you what, the red flag was kind of that so many different people on the internet, most of them with no qualifications, no more than I did, came up with such different things. I mean, there was a newspaper writer in Chicago whose expert said it was a possum. Somebody said it was a pig. Somebody said it was this, that, and the other. And so I began to think that, maybe they don't understand how these databases are constructed and what you have to do to ferret out the answers. So I looked more deeply at my own results, and I came upon the fact that all the polar bear data was in a totally different database, and polar bear is closer to black bear than panda, and I all of a sudden got some really good hits. There's very little black bear data in the databases, unfortunately, and so you have to go with other similar species, and that's not recognized by everybody either. So, I'm sure Dr. Todd knows that, but so I started getting better hits to a bear and to a human in the second sample and to a dog in the third. And in order for these to stand out, I invented these slightly different metrics, which Tammy alluded to with the equations that kind of make some of these hits rise to the top of the hit list a little better, because it can be a confusing mass of hits, all of which look pretty good sometimes.
So Dr. Ketchum what's the saying, Tammy? She was trying to make a silk purse out of a sow's ear?
That's it.
Maybe it was a bear's ear.
Dr. Todd, have you run any DNA sequencing from anything over the years in regards to Ketchum Camp?
So I inadvertently did. I have to reconstruct the timeline, but maybe 8-9 years ago, maybe a few fewer, I was actually given one of the samples that ultimately ended up in her study. One of the 110 samples or whatever that they allegedly sequenced the mitochondria of. But it wasn't connected to her. This was pre her involvement, and we genotyped it and got human mitochondria, and that's as far as we went. But the side story of this, why one of the reasons I'm still talking to you tonight, and well, I would talk to you any day, of course.
Well, thank you.
And Tammy, I'm not wearing my damn pendants. Shoot. I should have, I had it on earlier today, but when I changed my shirt, I took it off.
You have more jewelry than I do, Todd.
But Ketchum on her Facebook page, which seems to be her favorite means of scientific communication, accused me of being so disgusted that I got human DNA from this thing. She said that I destroyed the sample. And I have never destroyed a sample in my life. And so we were puzzling over this, and then we went back through our old emails, and we found the emails from the person who had originally brought the sample to my lab. When she asked for it back, we said, sure, come and get it. And I even have the dates and the emails of her coming to my lab to pick it up. I happened to be in Africa at the time, but my postdoc handed her the sample. And in fact, I even got her to email Ketchum to say, what you're saying on Facebook is completely false. I recovered the sample from, Disotell's lab, etc. And so that was, besides creating the journal, de nouveau, the lying about my destroying a sample because I was upset that it was human. I'm not upset with any result I ever get. Results are results, as Haskell can tell you. Whatever your experiment yields, that's what it yields. So that was a huge red flag for me as well. Besides the results from the original paper, and I really like what Haskell did, because I'm doing real science on this stuff all the day. I didn't have the time to go down and redo all of her GenBank searches and BLAST searches and all of those searches. And he did a wonderful job at that. I really applaud Haskell for your work on figuring out what those were. I mean, I had seen her results, lemur, gorilla, orang, all of these things all over the board, and they're what's in the database. And so if you're only looking, as he said, in a certain partition of the data, that's what you're going to find. And, the DNA continuity amongst life is so great that it's not shocking that a dog, a bear, a lemur, a coyote. I mean, Rictor, you are 38% identical to a banana.
Well, cool.
Parts of it might be.
So, and when you do these searches, again, you think, wow, 65%, 75%. It's not from 0 to 100. It's from 25 to 100. Because any two sequences at random are 25% identical. So you have to subtract 25% off whatever number you get just to start with. We share a couple thousand genes with bacteria because they're living, and life has, a single common ancestor, at least here on Earth. So when she had all these hits all over the place, yeah, some were human, some were other primates, some were unknown, a ton of them are bacteria. When we sequence, so they did two kinds of analyses. They did the mitochondrial analyses, which were all human, and then they did the next generation sequencing. But next generation sequencing, this new, it's just this marvelous technology that we're now using constantly in our lab. It generates tiny little fragments. You get 60 to 100, maybe 200 base pair fragments of DNA. And in the 3 billion plus bases of the genome, if you're searching for 100 bases, it's going to match every mammal out there. And then, as Haskell said, is it 98, 97, 96? Those numbers aren't actually that important. It's what it's embedded in, what it's surrounded by, and so on. So to say that, oh, it didn't match any known primate, of course not. So that analysis, her analysis is just the most superficial sort of one-off gloss. It's like doing a Google image search of a building, and there's a lot of buildings that look very similar to each other. And if you just grab the first one Google gives you without understanding the underlying basis for it, you're likely to be wrong.
Well, I wanted to talk about the paper a little bit to the people that haven't actually been privy to it, and there are links in some of the Facebook groups where you can go get a copy of this paper. Haskell has been kind enough to share it in the coalition. And where else is it available, Dr. Hart?
Squatch Unlimited, Team Squatch Unlimited, Melissa Adair's group. And I put it on the Bigfoot forum as well.
Okay.
And so far, 86 people have downloaded it. So that the word is getting out little by little. I've got some good comments back too.
I'll include a link to it in the description when the show's on YouTube so that people can download it as well. And I'll put that on the screen. To download Dr. Hart's paper, check the show's description.
Yeah, we could also put it as a file, a document file in the After Hours group too.
I think it might already be there.
Is it? Okay, great.
So. Yeah,
okay, so the introduction to your paper basically summarizes what's going on in the DNA research for Sasquatch, to prove the existence for Sasquatch. And it says that to date, no holotype specimen, which is normally required for the recognition of a new species, has been produced, okay? So this means that we don't have a body or something other than samples like hair, saliva, I think there's a toenail somewhere, but we don't have an actual body part. There was the alleged Bigfoot steak, but I think that didn't pan out.
Dr. Sykes proven that's fair.
The analysis is based on all of these specimens that have come in over the period of quite a number of years. I'm not sure what the earliest date is of the 110 alleged specimens that have been turned in. So we've got a lot of, probably old saliva, old hair, old shit, all of the things that Todd mentioned that are samples being turned in. So what is the chances of getting a good, accurate DNA test off of these kind of samples?
Well,
wouldn't that depend on how they're collected? I mean, there's contamination, right?
Exactly. There's so many things that circumstances that can affect these samples, how they're stored. I mean, we've seen that before where somebody's had a hair sample that they've collected for a number of years. How is that going to change the results whenever a test is done on these samples? And either one of you could jump in and answer this so that we can kind of get an idea of how these samples have been being tested. It's not really a uniform type of collection. It's not like a controlled environment that these specimens are being collected.
It's a very dirty way of collecting evidence.
Yeah, so can I jump in here for a moment?
Yeah.
Sure, please do. I was going to defer to you anyway. You're more expert in that.
The new technologies that we're using, and these are just years old. So stuff we did 10 years ago is like Stone Age. I mean, what we can do today is, we've sequenced whole Neanderthal and Denisovan genomes from 40,000-year-old things. We've done complete mitochondrial genomes from 400,000-year-old bones. So, and the contamination, it's a real issue, but we know what human variation is like, and these new techniques can gather so much data that even if it's 99% human contaminant, if there's 1% real something else, we can tease that 1% out now. Now, that being said, these techniques aren't cheap and free. They take a lot of effort and a lot of brain power and computer power to sort them out. So, people like me or Dr. Sykes and others don't want to just, everybody who says, oh, I found a hair, or I got some spit off a window pane, or I got this or that. We're not going to do every sample some squatcher claims is from Squatch. I mean, we do the first pass usually, and again, mitochondrial DNA is easier to get because there's about 1,000 times more mitochondrial DNA in any cell than there is nuclear DNA. So usually we do a first pass of mitochondria, and if it turns out to be horse or bear or human or coyote, I'm not going to go spend a lot of time and effort and money on doing further analyses, because you have to come up with a credible, why do you think this is Bigfoot? Show me the photo that's not blurry, the bone that's not a deer skull.
The follow-up examination at the location where the evidence was found?
Yeah, show me how you collected it. Because, if I know it's 99% contaminant, that's a lot harder to do than if it's 50-50 or 1% contaminant and 99% real. So, but again, these new tests aren't cheap. I mean, we're talking a few $1,000s, not 500,000. Don't know how they spent that much doing that study. We've sequenced multiple entire monkey genomes for a few thousands of dollars. I mean, literally $2,000 to $3,000 to get whole genomes from monkeys. But my lab is now regularly working with 100-year-old museum skins and hair samples and stuff like that. So if you could convince somebody credibly that this really is the magic sample and put a stake in it. I mean, I like to charge people, charge production companies, because I don't want to waste my time. If I just said, send me your samples, I would have 20,000 samples next week. I'm not going to do them. I need a stake in the game. And
it needs to be worth your time.
It needs to be worth my time and effort, because it's not insignificant effort. As Haskell can tell you, collecting the data, extracting the DNA, running the analysis is actually the shortest part of it. It's the computer searches and the analysis that actually take more time and effort than the few minutes in the laboratory setting up the experiment. Most of these experiments, a few hours of lab work, you put it into the machine, and days later you get billions of pieces of information. It's sifting through the billions of pieces of information that is the hard part, not the few minutes soaking it in a buffer, incubating it, vortexing it, running it through a centrifuge, and loading it into the machine. That's the easy part. It's sorting through the masses of data, and that's what Haskell did so wonderfully.
And that's where the Ketchum paper is lacking in the ability to do the sorting. That's what it gets down to, right? The bottom dollar is that the information that she had is not being processed correctly.
Exactly, and it's rookie mistakes. Can I call it that? I mean, I know she's not a geneticist. She, I think she even somewhere says she's a self-taught geneticist. That's fine. Clearly, Haskell taught himself far more than Ketchum learned in his reanalysis of her data, and he did a really nice job. It's just a job that I didn't have time to bother. I predicted it on multiple podcasts and interviews. I just didn't sit down and do the sort of slow grunt work, because I have real data to analyze every day.
Okay.
Well, and if I may say, I think Todd is wise to spend his time with the laboratory that he has, looking at real samples, controlled samples, and real data. And I think that I'm doing all I can. I don't have a laboratory, and there's really two steps here in analyzing all the data that comes out of the machine, and that is the first one to come up with the sequence, which I took face value and went from there. And I'm not prepared to say anything about the validity of her protocols or anything. I'm just looking at the sequence she came up with, and I think she must have used fairly good protocols, or she would have gotten garbage, and I wouldn't have found anything. So I think that part might be okay, but I can't believe on a paper with 12 authors that nobody would sit down and look at that final sequence and say, what is this really, and are we missing something? And it's just a mystery to me. I'd never let something loose like that if it were me, without a lot of cross-checks, especially on such a remarkable claim as this.
What about the fact that the name Homo Sapiens Cognatus, am I saying that right, was granted at Zoobank, even though an assignment of that name doesn't mean that it recognizes it as a species that actually exists, because there hasn't ever been a specimen brought forth. What good does it do to have a name in the Zoobank of whatever Homo Sapiens Cognatus means? I think there's a definition there that I don't know about as far as the meaning of the Latin of that word. How come they recognized it with crappy paper?
Zoobank is not an official entity. It's not, the IUCN. It's the Wikipedia. Literally, well, figuratively, sorry. I hate how we always misuse that word. It is figuratively the Wikipedia of names. It's the crowdsourced, open domain, public access version of naming stuff. So just because the entry is in Zoobank, it has zero scientific it's useful, just like Wikipedia is. I mean, I look in Wikipedia a dozen times a day to get a quickie first pass estimate as to information, but it's only a first pass estimate. And so Zoobank is not recognized by the international, it's not a scientific entity. It doesn't have scientific validity.
Kind of like De Novo Journal.
Well, it's better than that. It has multiple people, and yeah, let's just say it's much better than that.
Okay. All right. So, earlier whenever I said that, there's data there, but the processing of that data was, as Todd said, had rookie, what did you say, rookie mistakes?
Yeah.
Yeah.
So the basic principles, and it says this, I'm reading this right out of Haskell Hart's paper, the basic principles of nucleotide database search match were not followed. Right? The basic principles. So, my next question is, is the paper a blatant lie? Dr. Hart, I'd like you to answer that question.
Damn, Tammy, you just go right for it.
Okay, you're referring to the Ketchum paper, not mine.
Yes, the Ketchum paper, not your paper.
Well, at first I thought, well, she's doing her best, and unfortunately, I don't think she got the best advisors and co-authors that she might have. But then the more I read, it's like she's already decided they exist because she's seen them in her backyard, she told me, and in trees and so on. And I think she'd probably do almost anything to prove that, but it's hard to say. I can't say she was dishonest. Other people have said she's withheld some information that's key, and I'll let them speak for themselves on that. I don't think she's been forthright in everything. I wouldn't call her a liar or a faker. per se, but it's certainly not the standard of openness and honest discussion and willingness to admit that you might not have the answer or you made a mistake that I like to see in good scientists.
Well, she's also not coming in defense of her work. We have invited her on this webcast, Dr. Disotell and I. Tammy, we've invited her, all the joking and the the teasing aside. Seriously, let's come and discuss it. Well, you know what? Let's make a public appeal to Dr. Ketchum. Dr. Ketchum, you have provided us with so much material the past couple of years. Let's be honest. It's been fun, and we've had a lot of discussion regarding your hard work and results. Maybe you can come here after hours and be able to address some of the critics.
Oh, I would love to be on a show with her.
All right, so this is a public appeal to Dr. Ketchum. You're invited to come here on a webcast to discuss this with Dr. Disotell, with myself. I won't be bitchy, queenie, gay.
And Dr. Ketchum, I apologize for some of my earlier terminology.
Well, there you go. So, yeah, it'll be a great open forum where we can discuss this.
I was just upset over a couple things, and so some of my earlier terminology wasn't polite nor scientific, but I will absolutely have a clean scientific debate.
And we will be talking about a subject that we all love. We love the subject of Bigfoot, whether it's real or not, a monster, myth, angel, whatever it is. Let's all just talk about it, and let's give Dr. Ketchum the opportunity, the chance to tell her case. What do you think? What do you think there, Steve? You think that'd be cool?
I think that's a great idea, and I think Dr. Ketchum would be silly if her study is real to pass up this opportunity.
And Tammy, will she get a Bigfoot pendant out of it?
Yes, definitely.
And I know the guy who can do...
Dang!
Oh, yeah!
Oh,
wow!
Just saying.
All she had to do was say, no, thank you. Instead, what did she do? She goes off about how she lost all respect for Dean Cain for being on a Bigfoot reality show that is out to hunt Bigfoot, blah, blah, blah.
Yeah.
He's an actor. He's paid to be a host of a television show. He is not a person that takes Bigfoot as serious as we all do.
Yeah.
So she ignored us.
Lost all respect for him?
Yeah, she lost all respect for an actor, Dean Cain.
Yeah.
I'm sure Dean Cain lost a lot of sleep over that.
Yeah, I'm sure he's hurt.
She's been on other programs. She did an extensive series of radio interviews on the Steve Bradshaw show, which is in Nashville. I can't ever get him to come up on a computer, so I may ask him for a disc of those to really hear her side of it. Of course, he had no other people on to counter her arguments or anything, but she's willing to do interviews as long as they're friendly and,
Well, Tammy and gentlemen, if you recall, she jumped the gun in talking about her research and work to the media when Finding Bigfoot was going to have their DNA be looked at by a doctor, if you recall. So she wanted to, it was like a competition, a race to come out with information first with that television show, Finding Bigfoot. Do you remember that, Tammy? I certainly do. It was like an opportunity for her to come forward with all this information based on what? What about the peer review? What about the journal? You know what I mean? So that was the huge red flag in regards to me. I mean, if she had the goods, seriously, she should have kept her mouth shut instead of seeing that as an opportunity for fame, for look at me, I'm Dr. Ketchum, I'm proving Bigfoot's real. You know what I mean?
Mm-hmm.
So yeah, I would love to just have a sit-down, one-on-one discussion with her. I do this all the time. I have many colleagues that we, disagree with on various scientific, things, but we still have real, valid intellectual debates. We do it all the time for our grad students. We have the different people in my department or in New York City sit down and argue. I mean, we literally argue, but, civilly.
It doesn't come to blows now, does it? You guys beat each other up?
No.
Verbally, right?
So I would be more than happy, but I am, as Haskell mentioned, she hasn't followed protocol, so to speak, in the scientific method, et cetera, and she said some, she disparages her critics literally falsely, and in this case I mean literally, with actual proof. I remember one of her comments about me was, well, he's a mitochondrial DNA expert, so he wouldn't know anything else about the rest of the genome. I've sequenced multiple genomes of primates. 15 of my 70 papers are mitochondrial. The others are the rest of the genome. I know this stuff. I was trained in this stuff. I do this stuff on a daily basis. I just want to see the record set straight. And this story of hers is just very, very easy to debunk. She sequenced a bunch of random samples of a bunch of biological material, but it's not mysterious, unknown. It's not Bigfoot, angels, aliens, whatever. It's what you would get in sequencing almost any biological sample.
Well, I can tell you that I have heard her respond to her critics on another radio show with her comment, and I think this is pretty standard, of lies, lies, and more lies. That's pretty much how she responds.
Yeah, well, that's not how scientists do it. I've been mixed up in many a debate through the journals and at conferences and stuff. And you can't do that. I mean, and you can't create your own journal and claim it's peer-reviewed. This doesn't happen.
Definitely not a product of ignorance or negligence, right?
And Haskell raised a really interesting point earlier, that there's a whole bunch of co-authors. Where are these people?
Why have they been silent?
You're a co-author on a paper, you have equal responsibility for it.
The burden of proof.
In real journals, every co-author has to sign a document saying what their role in this is, that they stand behind it. We have seen Nobel Prize laureates have to resign positions because their underlings falsified data and published stuff that the Nobel laureate was a co-author on, and they had to resign positions because the data was falsified. If you're a co-author, you have responsibility for every word in that paper, as Haskell, I'm sure, knows in his career.
Well, I totally agree, and in that regard, I've contacted some of these co-authors. The microscopist said, I asked him why he might have found single-strand DNA in there, what possible reasons can it be, degradation or gene splicing or whatever, and he says, well, I only take pictures, I don't interpret cryptozoology. And I've sent my paper to as many of the others as I could get an address for, and I've gotten no response. I think they need to know what other people think of it, and I'm with you, Todd. They've been awfully quiet. She sent out samples to a lot of these labs, and I'm not sure they all bought into the final paper, to be honest. I think their name was put on before they knew it.
I have seen that happen, and I've tried to follow some of them as well, searching the publication databases and stuff, and I can't find some of those people. I know some of them are at commercial labs and so on, and it was a commercial lab that actually did, she does not have her own genetics lab. So I know that it was outsourced to various commercial labs to actually collect the data. And those people don't necessarily publish, so they might not be in the PubMed database and other things like that. But I would really like an accounting from some of those co-authors as to their roles and what they think, and did they approve of the final product? And more importantly, did they approve of putting their name in a fake journal?
Yes, I second that.
So you guys are all saying that this is highly unusual for co-authors to say nothing.
Yes.
I
Yeah.
have my opinion about it. I think that it's out of embarrassment. That's my personal opinion.
Well,
they're hiding because they're embarrassed.
Then you do a public retraction. If your name was included without your permission, you state that publicly.
Exactly.
But I haven't seen any of that. But to be honest, I don't like surf the Squatcher sites constantly because I would go insane.
Yeah.
Well, Dr. Todd, you are already a part of Zen Yeti. Mm-hmm.
Yeah.
Okay. Tammy, do you think that Dr. Ketchum is guilty of scientific misconduct?
Oh, yeah.
You do.
Mm-hmm.
For personal gains, for wanting to become famous, for
Plus,
wanting
it's
to...
not nice. And she tries to come off as Miss Nice. Yes, you do, Melba. And it's not nice to mislead people or to fib.
Where are the receipts? Where is the lab reports? You know, a copy of her data. Why isn't that being shared with everybody and everyone? Where's that?
So one of the things that has puzzled me, and again, I try not to waste too many brain cells on this. I have other things I would rather waste them on.
Diet margaritas?
So this was a $500,000 study, evidently.
I bet there was some party in there. What do you think?
Just, I mean, having paid for these analyses or carrying them out in my lab, I see the low tens of thousands of dollars of actual laboratory work. So, you know, there's always salary. You know, were these exorbitant salaries? Did they pay people way too much to carry out lab work, etc.? But I mean, you can do the amount of data produced is in the low tens of thousands of dollars of work, and yet this was 5 years and $500,000 of work. The math to me doesn't add up.
If I gave you $500,000, you could give me one hell of a good DNA study then, right?
Oh,
with the most modern testing.
Yes,
And
absolutely.
have money left over for margaritas.
Absolutely, plenty.
Dr. Hart.
Yes.
Did you submit your paper to her de novo journal, and what was the response?
Yes, I did. I thought I would face this head on, and I sent her a paper. It was slightly different than the one that you got. I've since improved it. And I got no response for a couple months, and I asked her about it, and she told me she had a hard time finding reviewers. And then I withdrew the paper and improved it and sent it to her again and said, do you have any reason why I should not try to get this published? Is there anything I'm missing? Am I wrong? And we talked for over an hour about a lot of things, and finally she asked me not to publish it, and I didn't press her as to why. I said I'd think about it, but then again I went ahead and submitted it to an online journal, which would have been one of these in which the user or the reader does not pay a subscription, and I would have paid a fee to do that. It is reputable, but they sent it out, and you saw the two reviews in my preface. One was very complimentary. The other was really, I thought, unfair, but I mean, that's just my view. in that he or she demanded that I apply my methods to something real, not a crypto species. And so I never got it. It never was accepted. And so I thought, what the heck, I'm not going to fight that. Maybe they have a valid point. I'll just put it out there and let some other people comment on it, and I'll learn some things from them. And hey, I'm retired. I don't need to build my publication list anymore. So I've gotten a lot of good feedback, and it's been very enjoyable, including this discussion, by the way. And so I'm glad with what I did. But I did submit it to her initially. I thought I'd just test, is this a real journal, and would she be fair to me like she wanted other people to be fair to her?
Well, because you have been one of her supporters, did Dr. Ketchum give you access to the rest of her data or just snippets of her work?
I got nothing. I did not ask for it, to be honest. I paid my $30 to get online access to the journal at first. Now she's put it free on her new website, but I paid my $30 just to have access and read it and get the sequences, and but I never asked for anything more. She brags that she has 4 terabytes of data, but I think she should share that if she thinks that it proves her point, you know, and even in supplementary files or whatever, you know. So that's not going to happen, I don't think, and I'm not going to ask her for it. I wouldn't know what to do with all that probably anyway.
The data she submitted, though, that's available, it's adequate to do the kinds of analyses you did, and to show that her results and conclusions just are not valid. So, I mean, yeah, you could get the raw trace info straight from the machine, but it's not going to change the result.
No, and I, as I admit, I'm not trained in that, so I wouldn't know what to do with it. I could learn, but it wouldn't be worth it. I just took her final result, you know, and even that was difficult and time-consuming to deal with. So I'm happy with what I did. I don't think it's the last word. I'd like other people to comment. I'm open to all kinds of alternate suggestions, whatever, and we'll just see.
Gentlemen, can you describe for our viewers what your own experience is with publishing peer-reviewed papers?
Today I spent, between soccer games, I spent a lot of time working on both the revision of a manuscript that we've had accepted with some minor revisions, and then another one that's been accepted, and I'm just doing the page proof, so editing all the little tiny errors. You know, that's what we do 5-10 times a year. And, you know, some reviewers, as Haskell found, you know, you'll get 2 reviews, one very positive, one negative, or usually you get 3 to 5 reviews, and trying to match all of their comments. But I wanted to mention something that Haskell said, and that is that the one was positive, and the other was like, it's fine, but it's not real. You know, and I have heard my friend Meldrum talk about this. You know, he has great difficulty publishing in the mainstream scientific literature some of his stuff, and you know, when at the conferences he's only allowed to sort of speak or show a poster every couple years. He's not there every single year at, you know, the conferences. You know, they let him do it every 2 or 3 years, and he puts on a good show then. But you know, I both understand the frustration of that. But then again, for a species that has no data to date, there's a really high bar that you have to cross to be accepted scientifically. So, you know, one real good DNA sequence, one really good picture, video, or bone, you transform the game.
Right. It
There's
changes the game entirely. Yes.
0 to date.
Right.
0.
I totally agree with that, and I thought my paper confirmed that, and that there still is no evidence. But let me just say a word on peer review. I'm in favor of it. I haven't published as many papers as Todd has, but I've published some good ones with a Nobel Prize winner, and I've published some on my own. And we do need peer review. I think it would be good, though, if some changes were made. I think the reviewers should consult with one another. I've often had opinions that I changed when somebody talked to me. And if everybody is well-meaning, I think that would resolve some of these huge discrepancies in evaluations on the reviews, and a couple other minor things. I think the reviewers should be more accountable. They're kind of, nobody knows who they are, and except sometimes you can figure it out. But it could be improved. But even in spite of my disappointment here, I'm certainly for maintaining a system, something like that.
Yeah, I mean, my favorite line about it is, I think it was Winston Churchill, you know, it's like democracy. It's the worst system of government except for every other.
Right. Or the alternative is much worse, or whatever. Yeah. Absolutely.
So, yeah, peer review, I mean, scientists are human. I mean, they have their prejudices, they have their likes, their dislikes, their lusts, their hates. Some of them have financial interests. They're human and do human things. Humans are really stupid. But, you know, we're still smarter than all the other things out there. Except maybe Squatch. He's stayed hidden for however long.
Okay, so being that Tammy and myself are not scientists and do not have a degree or any background in this field, okay, let's pretend I have a latex glove on, okay? And I have collected this French fry with the GPS positioning, a notepad, photographic evidence, date and time, the whole thing, written in a journal, and it's then put into a sterile test tube, okay? With the little face mask on, total sterility as possibly can, and I have this sample. I then send part of it to Dr. Hart, and then send part of it to you, Dr. Disotell. And I don't say what it is. I just say, would you mind doing this as a blind study? Tell me what it is. Okay? Now let's say, for the sake of argument, this is the Bigfoot French fry that's inside this test tube.
Yep.
And your results, Dr. Disotell, and your results, Dr. Hart, are identically the same. And you guys call me, oh my God, Rictor, what the F is that? Okay, where do we go from there? Are you guys considered peer review? Or do we send this sample off to other people and then get the whole scientific mainstream involved? What do you do from that point on?
So, if I may jump in here, I sort of have standing relationships with multiple colleagues that if I find a sample, I can send a test tube of DNA to a half a dozen people and say, test this for me. So if I had a sample that I was even remotely like, whoa, could this be it? Not, this is it. Scientists say, could this be it? I would immediately send that sample out for triple verification. If I had triple verification of that sample, I would then talk to the people who did it, because I'm not going to tell them what it is. This is a little tiny test tube of clear liquid. And they say, well, it's this, or here's the sequence. It doesn't, I don't know what it is, whatever. Then I would get really excited. But then I would have to ask them, you know, well, I want to go forth and propose it might be this. But I would ask their permission. Are you willing to be somebody who says, I sequenced this sample and got the same result. I wouldn't ask them to agree with my conclusion. I would only ask them to validate the raw data. So if the raw data was validated, and I've been here. I've been with new species and subspecies. So I have no trouble going off to peer review saying, I propose a new species or subspecies for this sample. And it's been the data has been validated by whoever is willing to say they validated the data. I mean, we don't want cold fusion. You know, we don't want some guys in Utah saying, look, we did this experiment and nobody else in the world ever validates it. Then I would look stupid. I would, you know, but if I find this thing, low probability, I'm out there. I will be the biggest, you know, fan of pushing this new thing. But I have to say. Now, let's take your French fry. I'm going to get multiple sequences from that. I'm going to have a bunch of 38% match potato.
And salt. Don't forget the sodium. There's salt contamination.
Well, that's not going to have DNA in it. I'm going to have some peanuts or sunflower or whatever oil it was cooked in. I'm going to have some Rictor. Well, unless you really collected it well.
I did, because Dr. Disotell taught me how to do it right.
Okay, so, but I'm also going to have some bacteria and some crap floating around the air and all sorts of stuff. So I'm going to have, if I next-gen sequence that, I'm going to have 1 billion pieces of data. And they're going to tell me all sorts of things. But very unlikely they're going to tell me unknown primate.
Yeah.
From your French fry.
So that was the reason I brought that question up, gentlemen, is because Bigfoot researchers out there right now banging against trees and making their whoops and calls and making their little forest friend bait traps, not realizing they're enticing bears into campgrounds, whatever. There's a whole ethical question mark about that. They're going about things the wrong way. For instance, when Justin Smea took pictures of the Bigfoot steak, he's holding it in his bare hand. You know what I mean? Human contamination right there. He didn't know different. He didn't know any better. Now he does, because Dr. Disotell, you have shown him how to collect DNA, you know, properly. So people, when they get their evidence that they think is evidence, to avoid future Dr. Ketchum circuses, don't send the entire thing off to just the one doctor, correct? Don't sign an NDA so that you're screwed with your hands tied behind your back. You know what I mean? I mean,
that's not science. I mean, an NDA is not science.
It's bullying. It's it arises a question of what's really going on here? What's the original motive? Money?
There's a big problem here in that, let's say you find your steak lying on a tree stump and you collect it perfectly and you dice it into 10 parts. There aren't 10 people out there who will analyze it for you. Unless you were, if you had the means, you could send those 10 subsamples to commercial labs with no more data, just like, I need a DNA workup of this tissue sample. But that's going to cost you some coin. So, you know, again, there's a catch-22, but the burden of, I won't say burden of proof, the burden of evidence is on, and I will become unpopular here, the so-called Bigfoot research community, because many of them are not researchers in the same vein that Haskell was and I still am. You know, if you're out there to prove a particular thing, that's not research. That's advocacy.
And you just hit the nail right on the head in regards to what everything has been in regards to Dr. Ketchum's DNA study.
Exactly.
Forest friends. Protect the forest friends. They're people too.
They're nothing. We got nothing on them.
And don't forget, Dr. Ketchum also said that they have constitutional rights, unquote.
Not in New York State.
Not in any state.
No.
Maybe in Washington, where they might be protected. If you shoot one,
Yeah.
you're going to go to jail. But
Yeah.
Just don't shoot one there. Shoot one somewhere else.
Shoot it, but drag it to Oregon.
So, but Haskell, what do you think about this sort of concept of? Do you think there are Bigfoot researchers out there?
Well, that's a good question. I'm a laboratory scientist. You're both sounds like a field researcher and a laboratory scientist.
Yeah.
I'm an amateur field naturalist, not a professional. And so these people, they're not using all the best scientific techniques, no. I mean, I wouldn't submit a sample unless I knew where it came from. I would like to see the animal deposited or have some idea. Just submitting hairs, I mean, that's just, that's too open-ended. And yet, they're doing the best they can. They do use some good technology as much as they know. And so I don't know what else. We're certainly not going to want to send somebody like you out there because your time is better spent in the lab doing what you're doing. And so I don't know that we have an alternative. I do believe it's possible that they may come up with something that's documented sometime. I don't think that it's an ape. If anything, it might be a feral human or something. I was interested in the recent work that showed that this woman, Zaina, who turned out to be a black African in Russia, and she's related to this guy Kvit, and so that what they thought was maybe some sort of a different hominin was actually a feral black African who had developed hair. And I've read these stories that kids get lost in Africa and they come back, they find them 5 years later, the monkeys have looked after them and they've got hair. Now, I'd like to hear your thoughts on that, but I mean, there is a possibility of some feral humans, but I'm not going to put any money on it.
Well, so I, in fact, way before Brian Sykes, who's a good friend and colleague of mine, looked at Quit and Zana, I actually sequenced their mitochondrial DNA, and I would have to look it up, but I did a show for National Geographic Channel, and he did it like 8 years later, and so he was able to like more fully describe it, but we found that Quit and Zana were related, they were human, and they were related to each other. I mean, the story was that she was a Neanderthal and he was her hybrid offspring, but no, they were both fully modern human and related to each other. I just, with the technology when I did it, I couldn't take it further than just human, whereas Dr. Sykes was able to take it a little bit further. But I did that same sample, and so the guy who gave them that sample has shopped that same sample like 6 different TV shows. I've seen Quit and Zana on at least 3 different television shows. And even when we say, nope, human, he still goes off and convinces some other producer. I've done the same thing with chupacabras. The Cuero and other chupacabras from Texas, I've sequenced those things. The Davis lab has sequenced them. Other labs have sequenced them. She keeps selling it to a new network, a new show. Oh, we need to find out what this is. Science is cumulative. Once you sequence something, you kind of know what it is.
And it doesn't change.
It doesn't change. But like the case with Dr. Sykes and I, he did it years later and was able to further refine which human it was. But I told him it was human way before then. But I worked on those very samples of Quittenzana in my lab. And I was just shocked when I saw them show up on Brian's show. Just like seeing Justin's bear show up multiple times. It keeps showing up over and over and over again.
So did you get to watch Bigfoot Bounty, Dr. Hart?
Yes, I did. Every single episode.
Oh, yeah?
It was enlightening. I learned a few things about the field research, how to do things, and how not to. And I think it's really big of Dr. Todd to participate in things like that, even though he is a skeptic. He's open enough to give other people a shot.
Oh, absolutely. And Bigfoot's fun. And there's a lot of, stories out there that you can't turn the blind eye to. I mean, as a scientist, the question is asked, is Bigfoot real? Well, let's find out. What do we have? What is being brought to the table?
My name is Dr. Haskell Hart. Thank you for being part of After Hours with Rictor. Come back next week for part 2. You'll enjoy it.
Thank you for being a part of this, Dr. Hart.
Oh, this has been very, very interesting and enjoyable.
A lot of unnamed sources outside the Bigfoot community has told me that Michael Merchant did, in fact, win the Spike TV contest.
Oh, shut up.